This procedure begins with labeling the DNA and polymer to generate a quantum dots or QD fret pair. Plasmid DNA is conjugated with quantum dots, the energy donor, and the cationic polymer is labeled with an organic fluorophore, Psi5, the energy acceptor. Upon mixing, QD fret nanocomplexes are formed via electrostatic self-assembly, and QD-mediated fret signal can then be followed as the interaction takes place. This QD fret system is introduced into a microfluidic channel, where the components will only interact through diffusion on the mixing interface. The DNA nanocomplex self-assembly can then be monitored by the combination of QD fret and microfluidics.
Hi, I'm Megan Ho from the Laboratory of Biomems and Single Molecule Dynamics in the Department of Mechanical Engineering at the Johns Hopkins University. Today, we will show you a procedure for combining quantum dot fret and microfluidics to monitor DNA nail self-assembly in real time. In our lab, we use this procedure to study DNA nail complexes for the application of gene therapy. So let's get started.
A biotin labeling kit from Mirus Bio is used to covalently biotinylate plasmid DNA with guanine-specific biotin labels, but scaled to have about 1 to 2 biotin labels per DNA. To begin, dissolve the desired amount of plasmid DNA into TE buffer to make a 1 microgram per microliter DNA solution. Set up the labeling reaction by mixing the plasmid DNA, DNase-free and RNase-free water, 10x labeling buffer A, and the Label It reagent. The Label It reagent is added last. Incubate the reaction at 37 degrees Celsius for 1 hour. After the 1 hour incubation, purify the labeled sample by ethanol precipitation following standard protocols. Store the labeled DNA at -20 degrees Celsius until use. The next step is to label the cationic polymer. In this demonstration, we will use chitosan as a model cationic polymer and label the free primary amines on the chitosan polymer backbone with Psi5NHS. First, calculate the required amount of Psi5NHS such that the molar ratio of Psi5 to primary amines is 1 to 200. This will facilitate complete conjugation of the Psi5 dye. Add sodium hydroxide to the chitosan solution in 25 millimolar acetate buffer to adjust its pH to about 6.5. While stirring, slowly add the calculated amount of Psi5NHS to the chitosan solution in a drop-by-drop manner. Cover the vial with aluminum foil to keep the mixture in the dark and agitate at room temperature overnight. The next day, purify the mixture by dialysis with a 10K MWCO slidalyzer from Pierce against 1% acetate buffer. Dialyze for 2 hours at room temperature in the dark. After 2 hours, replace the 1% acetate buffer and to dialyze for another 2 hours at room temperature in the dark. Replace the buffer again and dialyze overnight at 4 degrees Celsius in the dark. The next day, remove the purified labeled polymer from the dialysis cassette. The Psi-5 labeled chitosan is then collected in a microcentrifuge tube. And store at -20 degrees Celsius. Add streptavidin functionalized 605 quantum dots into the biotinylated plasmid DNA solution. Make sure you use the ITK series Q-dot streptavidin conjugate as opposed to the regular Q-dot series, as quantum dots in the ITK series are designed for the purpose of FRET. Incubate the solution at room temperature for 15 minutes. Keep the QD labeling reaction in the dark to prevent possible photo-bleaching. After 15 minutes, add the QD labeled DNA into 50 millimolar sodium sulfate solution to make the final volume 200 microliters. To prepare the Psi-5 polymer, calculate the required amount of chitosan for 10 micrograms of plasmid DNA according to the desired NP ratio, which is the theoretical ratio of protonated amines in the chitosan solution to the negative phosphates in the DNA solution. Dilute the Psi-5 chitosan according to the desired NP ratio with milliQ water to make the final volume 200 microliters. The QD-labeled DNA and Psi-5 chitosan solutions are now ready for loading into the microfluidic device, as will be demonstrated in Section 6. The silicon wafer is prepared beforehand by Purana cleaning and baking to remove moisture on the wafer. For the designated master thickness of 25 microns, spin coat the negative photoresist on the silicon wafer at 2000 RPM for 30 seconds. Soft bake the wafer on a hot plate with a ramp of 65 degrees Celsius per hour to 95 degrees Celsius. This will evaporate the solvent in the SU8 photoresist and solidify it before exposure to UV light. After the soft baking, apply a mask film containing the design of microchannels on the silicon wafer exposed to UV light at 365 nanometers for 250 millijoules per centimeter squared. Post-exposure, bake the wafer on a hot plate with a ramp of 65 degrees Celsius per hour to 95 degrees Celsius. Develop the wafer using SU8 photoresist developer. To check for complete development, immerse the wafer into isopropyl alcohol. The isopropyl alcohol should not turn milky. Finally, to reinforce the SU8 structure, hard bake the patterned wafer on a hot plate with a ramp of 65 degrees Celsius per hour to 200 degrees Celsius. Maintain the wafer at 200 degrees Celsius for at least 5 hours, then gradually cool the wafer down to room temperature. Place the SU8 master in a weighing boat. Mix the silicone elastomer and curing agent, polydimethylsiloxane, or PDMS, in a 10:1 ratio. Pour the PDMS mixture onto the SU8 master and leave the weighing boat in a vacuum desiccator to remove bubbles. Cure the PDMS at 65 degrees Celsius for 1 to 2 hours. After the PDMS is cured, peel the PDMS strip from the silicone master mold. Using a hole puncher, punch 2 channel inlets and 1 channel outlet for the fluidic device. Clean the PDMS strip and cover glass with ethanol and then air dry. Treat the cleaned PDMS strip and cover glass with oxygen plasma at 20 watts for 1 minute. Immediately bond the PDMS strip with the cover glass. Leave the bonded microfluidic chip in the oven at 95 degrees Celsius overnight. Plasma treatment and overnight baking are essential to enhance bonding strength. Before loading the reagents, fill the microfluidic channel with water to ensure there are no bubbles inside. This will ensure smooth flow during the experiment. Load the previously prepared QD-labeled DNA and Sci-5 labeled chitosan solutions into 2 individual glass syringes through the tubing. Connect the tubing with the 2 inlets of the microfluidic device. Be cautious not to introduce any air during the process. Set the flow rate at 20 nanoliters per minute under laminar flow conditions. Check the microchannels under the microscope. When the flow is stable, about 15 to 20 minutes, QD-mediated fret should be observed in the center of the channel. Acquire fluorescent images at different locations along the channel with a cooled CCD camera. Analyze these images with ImageJ and Origin Lab. Fret-mediated signal is monitored at different axial positions along the channel. Residence time is calculated based on the applied flow rate using this formula, where T sub r is the residence time, x is the distance from where the 2 streams meet to the position of the reaction under investigation, and v is the mean flow speed. The cross-sectional intensity profiles of fret-mediated Sci-5 signals can then be analyzed. In our system, the experimentally measured profiles exhibit a Gaussian distribution.
We have just shown you how to monitor DNA nanocomplexes through quantum dot-mediated fret in a simple microfluidic channel. When doing these procedures, it's important to remember to design a marker on your microfluidic channel so that the residence time can be estimated based on the applied flow rate and the channel geometry. So that's it. Thanks for watching and good luck with your experiment.